Hey everyone, and welcome back to Birds of a Feather Talk Together. This week, John, Shannon, Amanda, and I are joined by David Vander Pluym, a graduate student at Louisiana State University and Museum of Natural Science. Before beginning his graduate work, David spent years working along the Colorado River in Arizona and California, giving him a unique perspective on birds and some incredible landscapes. Today, David joins us to talk about his current research subjects, the Slate-throated Redstart, which may now be called the Slate-throated Whitestart. We get into this a little more in the episode, as well as a deep dive into its altitudinal migration. David was one of the LSU students who helped vote in our March Madness bird tournament this year, so it was a lot of fun hearing his thoughts on one of the tournament's biggest fan favorites, the club wing manakin. It might surprise you how he feels. Okay, let's grab our binoculars and let's get into it. All right, welcome back, everyone. So we're really excited to have David joining us.
Hey, everyone out there listening. I'm David Vander Pluym. I'm currently a graduate student at the Louisiana State University in the Museum of Natural Sciences. But before coming there, I spent more than 10 years working in field work with birds, primarily also along the Colorado River in Arizona and California.
So what made you go back to school after 10 years?
I always wanted to work with museums, and that was always a goal. And my now wife and I, we moved to the Colorado River for we'll be there for 2 years, and 2 years turned into 4 years, and then 5, 6, and it was got involved in some projects and with work there, and then it was kind of the project I was working on with elf owls ended, and it was sort of it's now or never before something new starts to go back to grad school. So I made that jump.
And you made the jump to Slate-throated Redstart.
Yeah, it wasn't. Have always really been interested in migration and bird movement. And then, what is it? In 2009, 2010, I worked for Alice Boyle studying white-ruffed manakins and altitudinal migration. I did along with my wife, we did the field work for her projects for 2 seasons, along with a couple other field techs. And I got really interested in altitudinal migration, which is more of people usually think about migration, they think of those long distance, just amazing things, from like the blackpoll warblers flying from New England to South America, bar-tailed godwits flying from the Korean Peninsula to New Zealand nonstop, and things like that with birds. But a lot of altitudinal migration is more short distance, tends to be up and down a mountain slope within the same mountain range. It's not always true. There's some ones that are amazing, like Baird sandpipers, which breed at lower elevations in Alaska, and then some of them will spend the non-breeding season high in the Andes. Or then Jessie Williamson, who's a friend of mine, her work with giant hummingbirds, which is just amazing work, and really cool, showing that they breed at low elevations in Chile, and then they spend the non-breeding season really high, and that has led to speciation as that kind of thing that really interested in, and partly just how these things are connected to each other, and why some are the same and why some are different.
What's it like to take classes again after so long, after being out of school for a while? Because I mean, people should know you can go back to school, right?
Definitely. People, if you want to, you can. I mean, it's been a weird experience, for sure. Different. Adjusting back to classes and things was interesting, but at the same time, it wasn't that big of a deal, to just get back in there, because it's always interested in learning, and expanding my knowledge base. So kind of being able to take classes wasn't really that hard, that big of a challenge. More of the challenge for it was just kind of going from I've been working on these projects, I have this whole network and this whole base of considered to be expertise of, and then kind of going back and starting from scratch. That was a little bit kind of at first of just being so this is what it feels like to start from square one again. But now it's, the museum is really, really a great place, and everyone is so collaborative and so helpful there. Yeah, it just made it feel like home, being there. Though that took a little while, because I also started during COVID, so there wasn't anyone really around. So it was that was also an interesting experience, but was good for me, because at the same time starting grad school, we had a kid, so it was actually really nice to have all my classes be online, and I could just have my newborn like asleep on my chest, and I'm like, yeah, I'm in class here, like learning stuff and everything. Just have the camera off.
That's awesome.
So you came in with this interest in migration and movements, and what was the process to get you to the Redstarts?
So that was really not knowing like if we'd ever be able to do field work, if the world would open up, and just kind of looking at what species were known to be altitudinal migrants, and then looking at what had a large series. And so it was kind of aiming at collections that had like common birds and things, and really thinking about what is going to be a common species. And so they're one of the more common species throughout the neotropics. So they occur from northern Mexico all the way to Bolivia and then out into the tepuis of Venezuela. So they're really widespread. But then in the literature, it also was some of them were known to be migratory, but most of them were thought to either be resident or just completely unknown. So I wanted to really, they're interesting to look at then if there was differences in the diversity that was based off of if they were migratory or not. Because you can get a lot of times with getting these species from migration is that you have a species was migratory, but then they, for whatever reason, decide, you know what, we're just not going to migrate, just be resident. And pretty soon they're now separate from that migratory population and you start to get a new species. And so that was something I wanted to try to disentangle. It's a lot easier to do the population genetics part of that than it is to determine if they're actually migratory or not, as they're so widespread. And the more learned about them, the more and more it was. And the more of my research with looking at them, the more it seemed like less at the like a taxonomic level and more at a population level. So it could be, oh, the subspecies is supposed to be resident. Except for this one mountainside in this one area. So that definitely makes it a bit more of a challenging, but also makes it a lot more interesting, in my opinion.
And can you, for people who aren't familiar with the slate-throated redstart, just describe the bird, how it looks, what compared to, and it kind of changes per region, right?
Yeah, that's part of the kind of cool thing. But so historically, they're thought to be closer related to the American redstart, and so they're pretty similar. Sort of in behavior and shape to them, though they have white in the tail rather than the red in the tail. And start is, I believe it's Old English for tail, and it comes from the European redstarts. And so actually a little side here, but there's a movement to change the names to being Whitestart instead of Redstart, because they're not closely related to American redstarts, and none of them have red in the tail, and only 2 species out of them have any red on them at all. But yeah, if you know like the painted redstart of the southwestern U.S. too, they're closer related to that, similar. So small warbler, like to flick their tails to try and flush insects. A lot of white in the tail. Then they have sort of a grayish, slaty head and back. And then in the north they have a red belly. But as you get into Central America and then South America, it goes from being this deep, vibrant red to then orangey red to orangey yellow to yellow.
Cool. And then do they have like a little cap too?
Yeah, they do. They do have a little bit of sort of a maroonish cap. And I'm horrible with like colored names and things. It's why artists make really good birders and researchers, because it's like they know all the colors to be exactly like, oh, that's this color. I mean, for me, it's, red, reddish, orangish. But yeah, that cap.
Although
all of them have it.
Go ahead, Shannon.
Its name, that pigment associated with its name, is actually really prized, apparently, historically, as the thing that makes that kind of deep, rusty, red, vivid. I don't know how to describe the color either, but the mercury sulfide that color, that mercury sulfide that creates a color like that, is kind of prized, apparently. So I found that was my interesting who knew kind of take on this bird. So that's where the miniatus comes from. It doesn't come from small. It comes from red. That red. Okay.
So David, when you're studying these, then can you just like big picture? I mean, how much of this are you going in the field to look at these birds? Are you mostly in the lab? I mean, is it a mix of both? Can you explain kind of what?
So for my research, because the whole project was designed during COVID, I will be able to do something without ever needing to go into the field. So I primarily have used museum specimens. Field Museum was very generous with donating both tissues along with actual specimens as well for this project. But I use museums for tissues from across the Americas, both North and South America, from various institutions, and then also the specimens themselves for looking at color, and then also doing a bunch of measurements on them. And that came from a lot of museums. I have gone out into the field for other projects, but then on this project, Costa Rica had only one tissue available entirely. And there's two subspecies that occur in Costa Rica, and there's some question if that should be two or if it's actually one. So I was able to spend a couple weeks down in Costa Rica to work on my project. And I mean, that's a horrible place to have worked. We stayed in these great
Exactly.
I was staying in like great Airbnbs, and us like hung out, and they weren't super easy to catch, but we were able to get a decent number of samples for this work, and so that was a lot of fun to be able to actually do what I kind of wanted to come back to work, back to the museum work to do, is to actually like get out into the field in someplace more exciting than the driest deserts of the United States.
Had you been to the New World tropics before you went there?
I have. I've just done. I worked in Costa Rica for Alice Boyle. That was spent about 6 months over 2 years in Costa Rica. Then I've also just vacationed to doing birding trips to Chile, a little bit of Peru before, and then Bolivia, also Argentina as well. And so I've done that in the New World tropics. I guess I got into the tropics a little bit in Northwest Mexico before, just for fun birding as well. But then, coming to LSU, got to go explore more of Peru, which was a lot of fun, and doing a couple expeditions down in Peru.
That's awesome. I was going to ask, you had 10 years of field work and being outside. What was it like to transition to like COVID era, where you were really just dealing with a lot of dead stuff?
Sorry, I need to get a drink real quick to answer that. I mean, it was what I wanted to do, but at the same time, I definitely miss the field. I'm a field person, and so I wanted and was hoping for that balance between them, because the museums are just amazing. There's so much you can learn, talk about. I regularly give tours, and we have the synoptic series, which is all of the species within that family, so that you can easily either compare or just kind of like show on tours. And just a synoptic series of tanagers, I've spent 2 hours on a tour, and it's just like we never moved. We just went through maybe like 15 drawers, and it was just so easy to just keep talking. I don't even study tanagers either, but a lot of people around me do, and you're such a cool group. So that kind of thing is just absolutely amazing from museums. You discover something every time you open a drawer. So that aspect is a lot of fun, and really interested in that. But then there's other parts, which is rewarding when you get the results, like doing the coding to do the bioinformatics and things like that. But the sitting at the computer and spending all that time doing it is not the most fun activity.
So
but
tell
the
us
results are
what kind
worth
of
it.
data you were gathering. Yeah. Tell us
So I've
what the
been
kinds of
doing.
data you were gathering and why you chose to do certain things.
So for this project, it's really wanting to look at the role that altitudinal migration plays in diversification. So the first part of it was kind of going into looking at what constitutes an altitudinal migrant. And so there hasn't really been a single clear definition. Different people have used it in different ways. Some people think it's just considered to just be strictly short distance. Others are like it can be long or short. Others are like it has to cross a certain threshold of elevational change. So first kind of looked at a wrote a perspective piece of what that is and what the terminology should be used for and how it has been used for. And then was able to actually start looking at what ones are altitudinal migrants. And the first was starting off with a literature search and just kind of finding all the information that was out there. And then I tried to use stable isotopes, so of hydrogen, and that's been used a lot in looking for altitudinal migration, but it's largely been used either with single locations or smaller bioregions. And this was looking at it from Mexico to Bolivia. And the way isotopes work is that you have this isotope of hydrogen called deuterium, and it's slightly heavier than a normal hydrogen molecule. And so the ratios differ because deuterium will fall out sooner. So closer to the ocean, you're going to have, or at lower elevations, you're going to have more of it. And so you can kind of tell where they're going, where the birds have been by looking at the ratio in their tissues and comparing two different tissues. But it's really, really complicated when you get to someplace like the Andes and you have these different watersheds that can receive just even slightly different rainfall and then how it gets taken up. And so wasn't really able to find anything with that was able to tell us much. Larger sample size, or then if you have stations right where you're collecting, that can be really, really useful too, because then you know sort of like this is the ratio that occurs here in this time frame, because it can vary year to year too, which can complicate it even further. So I turned to citizen science data and using eBird data to look for if you could see changes in the elevation. And so the northern one, Minneotis, is the one that's best known to be an altitudinal migrant. It has the most documentation in the literature. And then there's been a couple more that have known, but usually it's like one locality. They're known to be an altitudinal migrant. But using the citizen science data, I was able to see that Minneotis, the nominate subspecies, is an altitudinal migrant. You can see this nice percentage of eBird checklists reporting them from lower elevations occurs in the fall and winter when they'll be moving downslope and the non-breeding season. And then there's just no like records from the lowlands during the breeding season either. But for the rest of them, the data wasn't either not enough. Some of these subspecies, there are local, and there was only like 100 total observations, occurrence records for ever in eBird. But then was able to, on the flip side of that, the subspecies that's in Colombia, northern Ecuador, and into Venezuela, that one actually had the most eBird occurrence records. And that one was able to see this nice clear pattern of like, they're not moving at all. There's just no change throughout the year. And with that large sample size, feel pretty confident to say as a subspecies, it's not moving, but that doesn't mean that maybe there's a mountain out there where they do move, and they do move on a seasonal basis. Because that seems to be the case with these guys in a lot of places where just one area they will move.
So how do they interact with other members of the genus in the area?
For the most part, they don't really like overlap. And then during the breeding season, and that's partly of how they were thought to be like non-migratory, was in some places like Monteverde and Costa Rica. There's a lot of data, a lot of studies have gone, and they found that they can co-occur with the other species of Myobris that it's found at slightly higher elevations in the non-breeding season, but these guys don't move, they don't co-occur at other times. And so they're largely allopatric. During the non-breeding season, they all get along just fine, though in some places they do slightly overlap. And when we were in Costa Rica and trying to attract Minneotis, it would play and the other one would come right in too. And it'd be like, oh, you're the wrong one, go away. We want the other, your friend over there, not you.
Very cool.
So what else did you do? So you have the occurrence data. You tried the elements, and then did you measure color? You did genomics.
Yeah.
Clearly?
So
So what kinds of things did you do?
yeah, that was just one small part, was just determining whether or not they're altitudinal migrants. And so for the most part, I'm using the literature, but now I'll also have genomic data for all 12 subspecies plus the isolated population in the Tepuis is thought to be part of the subspecies that's found in the southern Andes in Peru and down into Bolivia. They look the same. They were indistinguishable, but there is no genetic data or genomic data for them. So I was able to get some and found that they were embedded. They were part of that group, which was really kind of cool to see. And I have the tissues I have sequencing for all 12 subspecies that have been described and are currently considered to be accepted. I just got back the last subspecies, though, less than a month ago, and I haven't had time to do put them into the pipeline. But the rest of them, so looking at populations of how they're related to each other, both with the mitochondrial DNA along with the nuclear DNA, and being able to see the relationships, and that there's three main groups. You have Minneotis is sort of on its own, and then you have a Central American clade, and then you have a South American clade, which is what previous work had shown as well. So it's nice to see that the previous work and my work lined up pretty neatly within the clades. It's not so neat lining up with the previous work, but the three major groups come out. And then I'm going to be doing different things, looking at if there's how recently they last came into contact with each other, if has there been any hybridization between the different groups, looking at how different they are genetically. These are largely all the different subspecies don't co-occur with each other. They don't come into contact anywhere. They can be where you can be on one mountain and look to another mountain and see a different subspecies, but they don't come anywhere into actual contact to look for if they hybridized or not. So have to sort of look at things with the genetics of how diverged they are. And then I'm also looking at the color using 6 different patches: the back, the crown, the forehead, throat, the chest, the belly, things that have been used to describe the subspecies. And then also have measured a lot of the kind of things that most people have measured: the length of the wing, the length of the tail, length of the leg, the tarsus, and then some different measurements of how wide the beak is, how deep it is, how long it is, to try to sort of see that there's real differences between them. And for most of them, there does seem to be some real differences between the different subspecies. But yeah, this is also really cool with the and I'm forgetting parts of my own projects. I got a lot going on with it, but also looking at the vocalizations for them, and then to see if they differ. They have a pretty simple song, but it does seem like it differs between some of the big genetic clades, the 3 genetic clades. So I'm going to be looking more into that. That is just starting to sort of do that work. And then also going to be looking at if they differ by environment and ecological factors. And presumably that they're not really going to differ any more so than that they're found from Mexico to Bolivia, which is a really big difference in habitat for what the species considered to be species is found in.
So what have you found that's been surprising to you the most so far?
Well, the confirmation that the belly color doesn't correspond to the genetic breaks has been really nice to see. That was previously published in a paper led by Jorge Perez Iman and published in 2010 or 2011, I believe. And that paper used a couple of mitochondrial markers, and that was sort of a surprising result from that. And then seeing it with both the nuclear data and the mitochondrial data, just that the color does not match where these genetic breaks are. And then finding in Central America was all the different clades really correspond biogeographically, which wasn't what was previously found. So it's just north of the Isthmus of Tehuantepec, that area in Mexico that gets to be a little bit narrow. You have the nominate Minneotis, and that's one clade. South of there, except for one population that gets just north of there, all the way to the Isthmus of Panama is another population. And then within that is broken up with like Northern Central America and Southern Central America. And the same is true with South America, where it's just really like the ones that are most closely related to each other are each other's neighbors. And so just kind of finding, sort of surprising that what we're finding is exactly what you would expect. Which is kind of interesting to see, because you a lot of times when you do this, you're going to find some weird or different results. And this has just been, other than the belly color, which is a separate story that I'm also working on as a not part of my dissertation, but that's a separate project, which I can talk about in a minute, a little bit. But yeah, just the finding with that really it's biogeography is really what separates these guys, which is a really cool thing to see. And then just how different they are genetically. That's not they all look pretty similar. But yet they differ really a lot with the mitochondrial, especially in the nuclear genome. It's between Minneotis and the rest is about it's more than 5% difference, which is usually with birds that's in the range of species. And yet some of these have been considered not to be identifiable to subspecies in the field, but they clearly know that they are different. And then the other surprising thing I would say is doing looking at the measurements, actually finding some differences. I was not expecting to find differences since these are all my arborists are pretty much the same body type, the same behavior, just in different areas. But tail length has really been very different between the different groups, with the birds in the north having a much longer tail than the ones in the Central America or South America, which is interesting too, because they're also the ones that are more migratory, and longer wings and tails can often correspond with that.
So I didn't know some of this stuff. The Jorge Perez did his research at the Field Museum, so we know Jorge pretty well at the time. And one of the things, I don't even know why I didn't know this, but I didn't, that I found fascinating was the experiments. So they took the different kinds of tails, Amanda, and went to Costa Rica and physically altered the tail to match the tails of the things that are further north and further south. And it turns out that the insects respond differently to the different tails, which absolutely blew my mind when I saw that. Amazing. And do you think that's going to hold true, like throughout the distribution, that, it matters how many spots or how deep the spots are, or how deep the white is on the edges of the wings? I mean, the edges of the tails?
It seems to. I'm not looking at the tail because they already did such a good job with that, of looking at the variation and the tail pattern between the different subspecies. But there's no kind of clinal variation with the amount of white in the tail. It's sort of these big jumps where it's sort of one will have more white, one will have less white. And then after those experiments that they did, those were only in a small area within Costa Rica, but it's just such a really cool result that it's got to be meaningful across their range, that this has to matter for some places. Maybe it matters more in like Central America, where you have a lot of these big changes going on between species, taxa that are found in like pine oak woodlands to being then in more like humid tropical forests, mid elevation. So those are going to be very different types of insects that they're feeding upon, and so would presumably respond differently. Maybe if they did this comparing something like the Costa Rica birds or the Colombia birds with like the Peruvian birds, they might find less of a difference there, but tail patterns still vary. So it seems like that's the most likely thing is that it's for foraging. Because they really, if you ever see them, it's really cool to see any of these guys, because as they're feeding, they're just constantly flashing their tail and constantly flashing that white and just showing it off. And then they'll flush an insect and you see them kind of like dive and with their tail spread and close, dive after and just chase after an insect and do a lot of like this little like tumbling and pursuing it. So it's a really cool thing to see. And so it just seems from the work that they've done, along with just how this species behaves and how all myovers behave, that it's meaningful in that way, as opposed to something else like sexual selection or just random drift. This one does seem to be real selection for getting food.
I'm trying to wrap my head around, how does the appearance of the tail relate to the eating of the insects? Did they use the tail to attract the insects, or did they use the tail to corral the insect? How does it work?
So it's what's called a flush pursuit. And so what it is that spreading of the tail and suddenly white, it's thought that startles the insects. And so then a lot of these insects are on bark, on leaves. They're really cryptic looking. And so instead of sticking your head and looking really closely or at everything, just going around, just constantly, suddenly flashing this white and taking away, flashing this white. And so the insects there's a ruby-throated hummingbird staring in at me. He got distracted there. I think it was picking spider webs off of our windows. But so yeah,
that's so cool.
The flush pursuit, it's thought that is trying to flush the insects, and so then that's that tumbling, chasing after that they do. So they scare the insect. It's like some weird, crazy predator that just suddenly showed up on my face, got to flee, and that is then they're able to be like, I found you, now you're my dinner.
That's awesome. That's so cool. So the tail is like a hunting dog for the bird.
Yeah, exactly. It's really like,
that's funny.
I'm just gonna flush everything. They don't have any pets that they can do that for them, but they do got their tails.
That's awesome.
That's so cool. David, I had a question for you, and I hope this isn't pivoting too much, but just get into your background and everything, have you always been into birds from a young age? I mean, at what point did you decide that this was something you wanted to study? I'm just curious kind of how you got to where you are.
I like to say I was born with a pair of binoculars in my hands. My parents actually met taking a college extension birding class in Southern California.
Oh.
So you hear a lot of people who are interested in birds, like kids don't necessarily get interested in it, but I was really hooked and really interested in dinosaurs as a kid. And then when I kind of learned that, well, birds are dinosaurs and they're still around as opposed to having to like dig up and look at fossils, I was pretty well sold on that and have been interested in birds ever since. Some of my earliest memories is being a very cranky 3-year-old because my parents didn't wake me up from my nap for a pileated woodpecker that flew over our car when we were on randomly, we were actually on a vacation to Louisiana. We
That's
grew up in
awesome.
Southern California, was living in Southern California, but I believe it was when I was 3, we went to Louisiana. Yeah, I was cranky about not being woken up from my nap for some new birds.
That's
Rightfully so.
awesome. Yes. Yes.
Yeah, I only held out against my
That is
parents
so cool.
for around a decade until I finally saw a pileated woodpecker.
Is that all? Boy,
That's
don't get on
hilarious.
your wrong side if a kid that young is giving a 10-year-old grudge. You,
But it's
You're
good because his
scary all of a
parents would
sudden.
get it. They're birders too.
Yeah.
They get it.
There also was a couple other times when it was close to a pileated, and then my parents got excited. They're like, David, and scared it away. So there was also that later on too. But now, I mean, I have occasionally we'll get pileateds just in our yard here coming by, so which is a pretty cool bird just to have randomly in your neighborhood.
Yeah.
Okay. So can I ask you, since we're talking about the pileated, where do you fall on the ivory billed?
Oh no.
Question.
Amanda.
Well, so I actually did the original surveys after Cornell announced them. And I spent 2 weeks in the White River, and it took me less than a week to be like, they're extinct.
Okay.
And yeah, my opinion has not changed since then. I sometimes not so delicately say I've seen better evidence for Bigfoot being around.
Okay,
That's certainly
that.
Bigfoot pictures are better.
Yeah. I mean, you can at least sort of see the details of like, oh, it does look like that's a guy in a suit. The surveys were a lot of fun, though. That was just a beautiful area. There was tons of woodpeckers there. I don't think I've ever seen better diversity or numbers of woodpeckers. Just not any ivory-bills left, sadly.
Okay, I feel like you sealed the deal for me now that I talked to somebody who was actually down there.
Yeah. We had, they're just, I was there in March, and it was sort of like this, we're going to go, they have them nailed down, there's got to be better evidence. And then it was just that the best evidence was in the paper at that time. And it was, they hadn't found anything else in the year. And I don't think anyone who was working that, I can't speak for anyone, but it definitely talked around and they're like, oh yeah, no, they're not here. I mean, they're not here this year. They weren't here last year, maybe, or something like.
Oh, okay.
But it's like, no, yeah, I
hear you.
Is your child interested in birds?
Bigfoot.
Sort of. He's interested in a lot of things. He's interested in the natural world, but Louisiana with all the mosquitoes, he reacts really badly. So he likes to go out and be in nature. But then it's like too many mosquitoes. We got to go. But he's interested in birds, but he also kind of is more just general liking things. I could see he has an interest sort of like in looking at plants too, or insects and everything pretty much. And but yeah, birds, maybe they'll come along. We're not trying to force him on him or anything, just kind of be introduce him to everything. And he really likes animals. And he really, really likes all animals, but he is definitely drawn towards us. A lot of the kids are those big predators and like wolves, tigers. He did one time tell me of like, Dad, you study boring things. Birds are boring. Study something cool like a tiger.
Well, our kid said bird watching is boring and you move too slowly. So that was his take on it.
Yeah, he has a bit of that take too. It's like, why are we still standing here? Let's go explore.
Yeah.
Let's go find a
Did-
bear.
Well,
Did you
maybe
say
he
your
won't
wife
have.
did? Let's find.
Oh,
Let's find a bear.
until he
Did you say
sees
your
that,
wife
then-
did field work with you too?
Yes,
Oh,
we've worked together on a number of projects. On the river, actually, I worked for her for a number of years, and helping her on her projects there, Colorado River. Whenever I say the river, that's the one I'm usually talking about. And yeah,
Very
so
cool.
she was a field biologist, ornithologist as well, and that's how we actually met. We originally met through the old like bird chat groups back in the early days of the internet. We did not care for each other at all then, but we met eventually at college, after college. And yeah, like as soon as she graduated undergrad, we just started doing field work together.
That's awesome. That's a good
Yeah.
story. It's a good love story.
And she's still actually, all of her work is still out in the Western U.S. With her. She works for Great Basin Bird Observatory, a nonprofit. So mostly working with the desert thrashers, Van and LeConte in Nevada and Arizona. So, but now a lot of it's remote, but she gets to go out and do field work a couple times a year to go play with the thrashers out there.
It's awesome. Awesome.
There's a couple birds we ought to talk about at some point.
I should
Yeah.
say, some desert thrasher sounds cool.
LeConte's thrashers, Bendire's thrasher, awesome birds.
Yeah, they're really cool, and they're steeply declining, and their populations just have really crashed in recent years. And so that's part of the work that they're doing, is that she's doing is they're tagged with satellite tags to look at how the movements, especially in response to solar going up, and she's found some really cool, really interesting results with that, with their movement that I can let her speak for.
Cool.
And then did you mention
What
there
is
was
your
some other project you were working on? Oh, sorry. Go ahead,
Yeah.
Shannon.
No, go ahead. I was just about to ask him about the red and what he was learning about the red.
Yeah, so another group had looked at sort of the colors of warblers and found that the same, looking at a couple color genes in particular, and they found that within Myoboris, the sister genus to them is Cardellina, which is like a Wilson's warbler, Canada warbler, but then red-faced, pink-headed, and red warblers that are found in Southwest U.S., Mexico. And those are the only warblers that really have this red on them. And then you have two Myoboris, painted and slate-throated, where they have red bellies. And they found that the same color gene, the alleles were the same for all the ones that were red. But then the yellow birds had different alleles, which matched the other yellow ones. And so Kevin Bennett then approached me and my advisor, Nick Mason, with working on this. And so we have combined forces, and now there's a group of us who are looking at sort of diving deeper into what's going on within the color of Myoboris miniatus. And it's seeming really interesting, where it is just the birds that are yellow just have this different allele than the ones that have the red. And that's not, and then from what they found, it looks like it's actually integration, where the Cardellina hybridized with Myoboris. And that's where it came from. But then you have these really deep splits as well. So
that's
it's sort of
cool.
then like, yeah, when did this happen and how did it spread? Because it's not the big genetic breaks is where you find that break in the color.
Yeah. So
Very cool.
why is color moving across the landscape differently than the birds do?
Yeah, that's something that is really interesting, and like we don't have a good answer for that yet. That's sort of like we just had a meeting this morning actually with it, where like talking about just the very preliminary results we've gotten, and so talking about maybe like future, trying to also look into that a bit more as well, because it's really weird and interesting there. Because it's definitely the break in color is between Honduras and then Costa Rica. So they're not really found in Nicaragua, which is an area that's much lower. So there's no habitat for them. And so that's where the change in that, going from sort of red-orange to an orange-yellow occurs. And yeah, somehow that seems to have has made it that far, but it hasn't made it down into like the South America birds. And yeah, just more work to do, more fun things to figure out and dive into.
Exactly.
And it's really I think it's just any group. You can pick any bird and there's going to be a really interesting and amazing story with them. And I just happened to pick
That's
this one.
what I think.
Yeah.
Did you vote in our March
I did.
Madness? Were you participating in that one?
Yeah,
Okay.
I did.
What did you...
Were you happy with the clubbed wing mannequin winning?
I think I was the one that made the comment about mannequins are overrated. So
Having worked on them, at least you had some
That's
first-hand
awesome.
knowledge.
because I've worked with
Yeah.
them, and then two of my office mates work with mannequins. So it's just been like surrounded by mannequins and a lot of mannequins, and everyone is just, I mean, mannequins, don't get me wrong, like mannequins are amazing. It's really cool birds. They are great, but there's sort of a sense of like they're the best in the end all of birds and things, and like they're cool, but I mean there's a lot of really cool birds out there. I do a lot of really cool things. I have
olive-sided flycatcher.
No, very thrush.
Yeah,
Like
I was
very
sad.
thrush.
I was sad to see Very Thrush go down. Just that song is just amazing with
Aw.
them, and yeah,
Next year.
yeah. There's a lot of just amazing birds out there, and I kind of also I like to root for the underdog ones too. So it's like let some of the other ones get some of the like fanfare, because it is a little bit of felt like club wing mannequin is the obvious pick.
Yeah. I have to admit, in the end, that was my choice. But I feel like I introduce people to varied thrush because most people have probably never heard of it. So
Yeah, that song.
maybe people will go try to find it in the Pacific Northwest. It's so beautiful. I mean,
There's such a bird of the
Yeah.
shadows too. A lot. I went to UC Santa Cruz for my undergrad, and we'd get them. That was sort of the southern edge of their breeding range. So there's like a few around that were breeding, but we mostly would get them in those like the big eruption years where you just get tons of them. And that would be really cool, where it's like this bird that you occasionally get a glimpse of in the like dark forest, more heard it, and then it's just like, oh, there's one hopping around underneath the bike rack.
Cool.
And that was just because
That's
there's
awesome.
just so many of them around. Like in those years, you could see like morning flights of them in the thousands of just going over. So it's just sort of like
Oh, wow.
they filled out all the habitat, and so they're just everywhere.
Wow,
Amazing.
cool.
for me,
Very cool.
seeing that bird, if you're in a place and you're actually seeing that bird, not just hearing it, but actually experiencing it, you know what it's like to be in the Pacific Northwest. Because to me, that bird, where it is in the environment, is just a complete immersion in what that place is like.
Very
Does
cool.
Slate-throated Redstart have a shot next year?
I mean,
David?
I love it. I think it's really interesting. And I mean, the stuff with like the color not matching with it is really interesting, like evolutionary. But as a species, I mean, I'll admit there's cooler ones out there. I'm like, it's not necessarily, it is. It's sort of like, as a whole, warblers are really cool. But they're colorful, they're charismatic. But there's other groups of birds that I really like too, and I find tend more
They're-
towards like being
they're-
my favorites.
Well, we
flesh
won't
and-
do another mannequin, so it won't have to be out competed by the mundane next year. John will nominate some other dull, boring thing with no color and no personality.
I'm going for southern whiteface.
Is that an Australian bird?
Yep, that's an Australian
Okay.
thing. There you go.
Okay.
Nice.
That's what you just saw
Next
those
year,
recently,
that's,
right?
That's my I'm gonna nominate that next year.
Very cool.
I will say with like all sided flycatcher, it's growing up in Southern California, that was like one of the birds of the mountains, and learning that early on of being like the quick 3 beers, and then you'd have mountain chickadees being like cheeseburger, and so it was just like, man, this is like the perfect place. Just to be up in the mountains. And so yeah, that's kind of the two species that really like takes me back to like hearing like seeing an all-sided flycatcher just makes me think like mountains.
My gosh. David, that cracks me up so much because RJ and we met in Colorado. We met in Steamboat Springs, Colorado, and we, of course, hiked a lot. And RJ's thing with hiking is like, the only reason I hike is so I can eat, like, afterwards, like, eat whatever I want. And he said, like, when I'm hiking, I'm just thinking about food. And I'm wondering, like, do those birds actually sound like that? Or is that just like...
They definitely sound like that, because I learned that.
They do.
Yeah,
Okay.
I learned that, and I heard them like long before I ever took a drink. Like, I mean, I learned that from being a kid.
Okay.
Um, so yeah, so
Okay,
it's
I
like
believe you
I
then.
did. It does sound like it. It definitely and like yeah, the chickadees going cheeseburger also. They definitely sound like that. Like
So they're not
That's
just
that's
awesome.
not.
frat boy paradise birds.
I mean, they can be both things. Like
Yes.
They can.
That's
I
awesome.
never thought of those birds in that way ever.
yeah, it would make me. I was just gonna say yeah. It's, sometimes instead of it being like I was hungry and that's why I was hearing them, it was be hearing them would make me like hungry and be like, yeah, you know, it is
Yes.
lunchtime. I'm ready to head back
It
to the
is.
field and stuff. Like
That's awesome.
Okay, so RJ, what's the best thing you've eaten after a hike then? If you dream about food while you're
Oh,
on a
I mean,
trail, what is it that you want?
it's usually a cheeseburger without cheese.
There you go.
A burger
And 3 beers.
and 3 beers.
Three
Quickly.
beers.
I heard a magpie one time say hot dog. No, I'm kidding.
There definitely is a little bit to like you can hear anything you want to hear because I remember there's a group of friends we would in undergrad we would do go out to the Mojave Desert of California looking for vagrant birds at some of these really isolated patches of habitat. And there's this one place that's just an amazing migratory stopover point called Thunderbird Springs. And that place, it's a couple acres of like cottonwood willows, but you could see several thousand birds in the morning just cruising up this canyon. And there's this one evening where we're just like sitting and we were drinking a couple of wheat beers and we realized that it's like, that's what willow flycatchers say. They actually go wheat beer. And it's like, they do not sound like that. But in the context of it, it's like, oh yeah, they totally sound like that. Like, definitely was able to make it into being that's what they said.
That is so
Okay,
funny.
so what's your favorite Louisiana-centered food? What is it that you like? If you go to a festival, what do you want? What is your favorite?
That's hard. Oh, that's really hard because there's so much that's really good. Probably a toss-up between something like crawfish etouffee or boudin.
Oh, those are
What's
good choices.
boudin?
It's a
What's
type
that?
of sausage that's really like rice-based with just spices and meat, and
Oh.
it's really delicious. And it's like you get it here, and usually the gas stations are the ones that have the best boudin.
Mm-hmm.
Oh, wow.
That's right.
And then
The
it's
gas stations
a-
and the rice fields.
yeah, because it is a Cajun dish, and it's the sausage coming from being that this was whatever meat they could get with all that plentiful rice they had. But yeah,
Very
that was
cool.
probably two of my favorites. Catfish too. Catfish po' boy is always good. Gumbo, and not this time of year, but in winter, it's always really good. And my wife
What do
is
you say, John?
gosh.
What is your wife like?
my wife's actually part Cajun, so she
Oh, cool.
has in the wintertime she'll sometimes make like gumbo and things, and then I'll make
Oh,
other learn how to make other dishes, Cajun dishes. So
that's awesome.
dive right in
Cool.
with it.
Man, I'm hungry now.
Yeah.
Sounds so good.
Cheeseburger.
So good.
Yes.
And 3 beers.
Yes.
So, John, what would you say your favorite thing in Louisiana was? What did you like the best?
Crawfish etouffee, hands
Yeah,
down.
I just like the
Shannon, what's
crawfish
yours?
itself. Yeah, the
Just the
crawfish.
crawfish.
Yeah, but I don't like potatoes, so that part I didn't care about. I don't like potatoes, but and then the other thing was they would have crawfish bread at festivals, and that compared to other festival kind of food, I thought the crawfish bread was the absolute best of those kinds of things I'd ever tasted. Loved it, delicious. But I actually like so many of those foods. You know, that Italian restaurant that used to make the crawfish pasta was so good. The shrimp po' boy at the Chimes was one of the best things I ate there. And that's just a bar / music place. At least it was when we were there.
Sadly, it no longer does the music, but it's still there. It's still like hangout for grad students going there, and that's my favorite catfish po' boy is from them.
Oh, that's awesome.
But yeah, we probably go
cool.
there too often at times. Some of the wait staff are like, no us on site.
That's awesome.
That's awesome.
That's a good thing.
Yeah.
That's positive. Yes. Yes.
Well, David, this has been so awesome having you on. Thank you
Thank
so
you,
much
guys.
for
This was a lot of
joining.
fun.
Yeah. This
Thanks
is
for
so
coming
much
on.
fun. Yeah.
Is there anything else you want to plug for Slate-throated Redstarts, or any way anyone can support your research, or anything you want to promote?
Well, I'm looking at graduating soon, so my sort of winding down this part of the research. But yeah, I mean, Slate-throated Redstarts are a really, even though they might not be my favorite, they're definitely one of the cooler birds out there. I think just supporting science and knowing that there's a lot of amazing work going on at museums is the best. Use citizen science and community science is just fantastic for that things, but definitely also read the best practices part, because that makes your data to be a lot more worthwhile as community science. Because in order to do my research with using community science, I had to do a lot of filtering, and a lot of that filtering ends up being then you have a lot less records to use, especially for some. So following those best practices, if in doubt, split up your checklist is always a good thing.
All right. Well, thanks so much for coming on.
Yeah, thank you all.
Thank you.
Do it again sometime.
This was fantastic.
Yeah.
Thank you so much.
Yeah. Awesome. Thank you.
Good luck with the end of your dissertation,
Thanks.
too. Not the easiest thing in the world.
Yeah.
Thanks so much to David for joining us, and thanks to all of you for listening. Next week, we'll be back with a brand new episode. So thanks, everyone. Enjoy the rest of your week, and happy birding. We love you all.